Hi, I'm George Hammond, Team Lead of Conference Production at Technology Networks, and I'm here to introduce our next talk from our Technology Spotlight sponsor, Twist Bioscience, identification of Dickkopf-1 antibodies utilizing a multifaceted discovery strategy. It's my pleasure to introduce Hua Gian, who will be sharing her expertise on this topic. Hua oversees the product portfolio of Twist Biopharma Solutions as its product leader. Most recently, she was the Senior Director of the Antibody Engineering Department of Twist Biopharma Solutions, leading the in vitro antibody discovery and optimization team. She has over 17 years of antibody discovery experience, primarily utilizing phage display technology for early-stage research programs. A very warm welcome to you. Following the presentation, we will also have a Q&A session, and we welcome any questions that you might have. Please ask questions using the Q&A system to the right-hand side of the video player. And be sure to check out all of the fantastic literature resources available from Twist Bioscience in the literature and resources section of the platform. And so, without further ado, I will now hand over to Hua to begin her presentation.
Thank you for the introduction, George. Good morning. Thank you to the event sponsors for the invitation, and look forward to speaking with you all today about Twist Cancer Research Solutions. As noted, Twist products are for research use only. They are not intended for the diagnosis, prevention, or treatment of a disease or condition. My talk today will be in 3 parts. We'll have an overview, discussion about cancer research solutions at Twist, and an immuno-oncology case study we identified here at Twist Biopharma Solutions. Twist Bioscience is a market-leading DNA synthesis company founded in 2013. We have a global presence, ensuring that our customers can always access someone local to support them with their work. We're headquartered in South San Francisco. Our manufacturing all happens in the US at our Factory of the Future in Wilsonville, Oregon. Our core technology is a silicon DNA synthesis platform that enables massively parallel synthesis of oligonucleotides using our proprietary chemistry. Additionally, we have invested significantly in workflows and processes beyond DNA synthesis R&D. We invested in our chemistry with our Twist Silicon platform. We're the only company who can make oligos at a consistent quality of up to 500 nucleotides. These quality oligos are then utilized for the construction of our product portfolio. We invest in experts in automation at the Factory of the Future. Our factory makes over 5 million oligos per day, and our experts are aided by robotics that convert them into DNA and protein products for research. We also have a world-leading team of DNA and antibody experts. Our in-house experts include leaders in research, drug discovery, and biosecurity. We also have a global team of FAS and technical experts who are on hand to support you with the design and development of your projects. Our researchers use our DNA and are writing the future: chemicals, food, therapeutics, and diagnostics. We've spent 10 years optimizing the oligos process, and it allows us to generate the highest quality oligos on the market. Unique amongst our high-throughput synthesis methods are our very low error, very high uniformity oligos that are used as a raw material for the generation for the rest of our portfolio. Here you can see our oligos are used for oligo pools, MGFs, and gene fragments, and our synbio products like our site saturation and combinatorial libraries, in addition to our NGS and biopharma portfolios. We like to say that quality raw materials in equals quality products out when it comes to our entire product portfolio. Onto our cancer research solutions. Our product portfolio is designed to support cancer researchers throughout the entire research continuum. We see this journey as comprising 3 stages: discovery, therapeutic development, and detection. In the discovery phase, researchers can use our synthetic biology and NGS tools to decode the underlying disease mechanisms and build accurate in vitro cancer models. In our therapeutic development phase, our biopharma portfolio helps researchers design, engineer, and optimize novel therapies, everything from antibodies to engineered cell therapies. And in our detection and diagnostics phase, Twist NGS solutions empower research into cancer biomarkers, liquid biopsy detection, and minimal residual disease monitoring. All of these tools work together to offer a comprehensive, seamless suite of solutions. Looking closer at our discovery stage, Twist tools empower researchers to deeply explore the basic biology of cancer, whether it's decoding genetic drivers or building better in vitro models. For those investigating functional genomics, we offer versatile solutions for popular research approaches like CRISPR screens and massively parallel reporter assays. If your work involves creating new cancer models, our synthetic biology portfolio has you covered with custom genes and constructs in multiple formats. And for researchers diving deeper into the cancer's genome, our NGS portfolio provides comprehensive sequencing options and span the whole genome, targeted panels, RNA sequencing, and methylation analysis. For detection and diagnostics research, Twist has NGS tools that are highly sensitive and support several different workflows. For liquid biopsy, we have a suite of tools that support MRD testing, therapy, matching, and early detection. We also offer RNA sequencing kits for investigating the transcriptome. Our exome panels can be used for genomic profiling. And lastly, our suite of methylation sequencing kits can offer insight into the epigenome. On the right-hand side here, you can see that we play in the library prep and targeted sequencing side of the NGS workflow, and our kits are compatible with several leading sequencing platforms. Our Biopharma Solutions group utilizes integrated in vitro, in vivo, and in silico antibody discovery services to help identify antibody binders and leads for bespoke projects. From custom antigen production through antibody discovery, engineering, and expression, we have all the pieces in place to make, test, analyze, to produce better lead candidates faster. Last part of this talk will be about our immuno-oncology case study. The Dickkopf-related protein 1, or otherwise known as DKK, inhibits Wnt binding to LRP56. The Wnt signaling pathway historically has been associated with homeostasis, but recent developments have suggested that it plays a role in oncology. As DKK1 is a soluble protein that competes Wnt in binding, our intention was to determine whether DKK1 plays a role in tumor development, and if so, if an antibody can be identified that works as an antagonist to inhibit tumor growth. DKK1 contains two cysteine-rich domains, otherwise known as CRDs, that appear to regulate Wnt in different pathways. CRD1 regulates a non-canonical pathway, which function is still unclear, but has been suggested to induce immune response, which is an advantage targeting a non-canonical pathway. CRD2 regulates the canonical pathway, which is associated in the canonical signaling associated with homeostasis. We wanted to use our library of libraries to identify binders that either bind DKK in CDR1 or CDR2 domains. Leveraging Twist core DNA synthesis technology, we within Twist Biopharma are able to leveraging Twist core DNA synthesis technology, we within Biopharma are able to generate high diverse libraries that can be panned against many targets of interest, including DKK1. We design phage display libraries with CDRs that match natural human repertoires, while other libraries have been generated by random sampling from desired sequence space. Using a combinatorial approach, we can very quickly make very high diversity antibody libraries for prosecution. Two of our libraries that we used in this campaign were from our VHH single domain libraries, and they were used for bead-based panning. We leveraged these humanized VHH libraries with varying CDR diversity to pan and screen DKK1. They were generated using a humanized framework with the incorporation of unique llama CDRs in the H-shuffle library, and in the H-shuffle HI library, 2.5 million CDR3s were shuffled in to make a very high diversity phage library. Utilizing these libraries, we use them in our in vitro antibody discovery workflow, where we utilize stellar B-phase panels to identify antibody binders, identify single clones using ELISA and Sanger sequencing, reformat N2 IgGs for expression and purification, and then further characterize using bindings functional studies. In as quick as 10 weeks, we can identify antibody binders that can be characterized and, if need be, further optimized. Using these library of libraries, we performed 3 campaigns where we used the VHH library series in one campaign, and additional 2 campaigns using machine learning approaches to identify a number of binders that we reformatted for primary assays and further down selected in functional assays and immune activation assays. A handful of these clones were shown to be tumor killer cells and were moved down into an xenograft study as well. Our first identification was to look at the different binding profiles of these antibody binders by epitope thinning. Different binding profiles were identified, with some clones showing triple cross reactivity, and some binding CRG1 only, and others clones binding CRG2. We focused our efforts to further characterize CRG1 binders while continuing to use a CRG2 binder along with a positive control in further studies. Using in vitro cell-based assays, clones were confirmed to either elicit the canonical or non-canonical Wnt signaling pathway. In the top panel, the antibody lead that was the CRG2 binder, along with the positive control, demonstrated that binding of these antibodies reactivated the canonical Wnt pathway and restored cell differentiation. For the CRG1 binders, immune activation was observed in the PBMC assay with increased activation of GM-CSF. To build on the notion that CRG1 binders can elicit an immune response that can lead to cytotoxicity, we looked at different cell lines to confirm that cell killing ability is due to our CRG1 binders. We're able to indeed show that these CRG1 binders induce cytotoxicity in numerous cells and cytotoxicity assays, giving us further confidence that our leads were indeed going to be effective in tumor killing. A handful of leads were used to test whether tumor growth could be inhibited in a xenograft study. We were able to identify that our lead TD725-003 showed statistically significant tumor suppression in this assay. We were encouraged by these results and have continued to further develop these leads, but are open to further investigation with others as well. We're very encouraged by this study to show that our fast, rapid identification of DKK1 binders using our libraries and our ability to improve using some machine learning models allowed us to identify antibody binders that are, in comparison, contending with clinical molecules. This gives us great encouragement that our in vitro antibody workflows, along with our library of libraries, can empower others to identify binders in cancer research as well. In summary, we believe that DKK1 activates non-canonical pathway and immune response, and that targeting DKK1 CRG1 is an advantage for cancer therapeutics. Our high diverse libraries enable the epitopic diversity that could lead to deciphering different biological functions, and our in vitro preclinical data support the inhibition of DKK1 as a therapeutic strategy for the treatment of cancer. With this, I thank you all for your time and attention and welcome any questions.
Thank you so much, Hua, for that fantastic presentation. We really do appreciate you sharing your expertise and insights with us today. And I have no doubts we are likely to receive lots of questions from the audience, so I will move straight onto the Q&A now. I can see. So your first question is, does Twist Bioscience offer any solutions for companion diagnostics?
Yes, we do. Our best solution would be our custom target enriched panels. We can have highly customizable NGS panels that can help track genomic alterations relevant to therapy response.
Wonderful. Thank you so much for that. And one follow-up question that we've had is, does Twist offer any regulatory approved kits?
Yes, we do. We offer a whole exome library preparation and target enrichment workflow with IVDR certification. We have a precision prep and enrichment diagnostic kit, a precision exome diagnostic kit, and an exome diagnostic panel as well.
Excellent. Thank you so much, Hua, for another fantastic answer. And following on from that, in your experience, do you see customers using NGS to characterize the tumor immune microenvironment?
Yes, it's possible to leverage NGS to provide a comprehensive view of both tumor mutations and immune cell signatures. We can also simultaneously analyze genomic, transcriptomic, and immune profiling, and you can get a better understanding of response to immune therapies.
Amazing. Thank you so much. I believe we have time for a few more questions before we close out this session today. And moving on to the DKK1 side of things. So your next question is, besides the TCF and LEF assay and the NK cell assay, were there any other assays performed to validate the WNT canonical, non-canonical pathway signaling?
Yes, we validated the binders using the phospho-junk ELISA assay, and that is part of the non-canonical pathway. And we performed mineralization assays and to confirm the canonical pathway.
Excellent. Thank you, Hua. And we have time for one final question today, which is, what are the next steps for your DKK1 leads?
So our DKK1 leads are open for licensing. They're also open for evaluation. So if there are any researchers out there or companies that are interested in pursuing DKK1 as an asset, we're willing and open to talking with you all.
Excellent. Those are all fantastic questions, and a huge thanks to Hua for such insightful answers there. Unfortunately, though, that is all that we have time for today. For those with unanswered questions, don't worry, we'll get them straight to the speaker after the event. And remember, this talk, like all the others in the event, will be available for you to revisit for the next 30 days. And whilst you're here, don't forget to check out the free resources from Twist Bioscience, which you can now download for free just below this live stream player. So that just leaves me to say a massive thank you once again, Hua, and to everyone in the audience, and we will see you all again soon for the next talk. Thank you.